Hello everyone. Today we are going to discuss about enzyme pattern in diseases. So in the previous classes we have studied lot about enzymes and what are the enzyme inhibition and what are types of inhibition, and today we are mainly moved into the enzyme pattern in diseases. So how the enzyme pattern will vary in some of the diseases and how should we diagnose based on the enzyme for respective diseases. So let's move into the topic. So as we all know, the enzymes are the biological catalyst which speed up the chemical reaction that accelerates the biochemical reactions in the body. And these enzymes are normally present inside the cells of the tissues and organs. When these cells are exposed to damage due to any type of diseases or due to any type of injury or infection, these enzymes leaks into the bloodstream. And whenever the enzyme leaks into the bloodstream, this measurement of these enzymes helps in the diagnosis and monitoring of various diseases. So different diseases shows different patterns of enzyme elevation, which helps the clinicians to identify the affected organ. So there are several types of diseases accompanied with the enzymes like liver, heart, bone, prostate gland. So the clinically importance of enzyme pattern is to detect the organ damage, to detect any type of tissue injury, and to identify specific disease conditions, to monitor severity and progression of diseases, and to evaluate the effectiveness of treatment based on the enzyme elevation. And it is very much important in cardiac emergencies, liver diseases, and pancreatic disorders. So some of the organs which is having enzymes inside the organ is heart. So heart, it is having an enzyme CKMB, that is creatine kinase MB, troponin, and LDH, lactate dehydrogenase. And in liver, ALT, AST, ALP, and GGT. So alanine transaminase, aspartate transaminase, alkaline phosphatase, gamma glutamate transferase. And in pancreas, amylase and lipase, both enzymes are secreted in pancreas. And in bone, alkaline phosphatase enzyme synthesized in bone. And in prostate, acid phosphatase is synthesized in prostate gland of men. And first we'll move into the cardiac enzyme pattern. So enzymes in myocardial infarction. So whenever the heart attack came to the patient or a patient affected by the heart attack or cardiac arrest, these enzymes gets elevated into the bloodstream from the heart itself because their heart muscles get damaged or blocked. So even though the muscles have been blocked, the enzymes released into the bloodstream. So first enzyme is CKMB. So this creatine kinase, which mainly helps in the energy production for heart muscle contraction. So this normal range should be 0.50 to 5 nanogram per ml. Suppose if any abnormal values of above 5 nanogram per ml, this leads to the specific indicator of myocardial infarction, that is heart arrest. And AST, aspartate aminotransaminase, it's mainly involved in amino acid metabolism and energy production. So this normal range should be 10 to 40 mu per L. More than 40, it indicates the heart or liver damage. And LDH, lactate dehydrogenase, it mainly converts lactate into pyruvate with the help of LDH during energy metabolism. So the normal range should be 140 to 280. Above 280 mu per L, it indicates the tissue damage, heart tissue damage. And troponin 1, it mainly regulates the cardiac muscle contraction. So below 0.04 nanogram is normal. Above 0.04 is the most indicator and specific marker for heart attack. So while a patient having heart attack or cardiac arrest or myocardial infarction, these enzymes serves as the key indicator for the diagnosis and monitoring of heart attack. And during and after heart attack these enzymes leaks into the bloodstream and it should elevated in the bloodstream for at least 12 to 24 hours. So this is all about the cardiac enzyme pattern.